Thank you everyone for joining us tonight. We have a topic by Dr. Xiang, which is lysine acetyltransferase, KATs, and developmental disorder. As you all know, Dr. Xiang is a PhD professor at Goodman Cancer Center, Department of Medicine at McGill University in Montreal, Quebec. He's also one of the world pioneers in studying the MOS gene. He's one of the very few scientists in the world who started working on the MOS gene many years ago. Welcome, Dr. Xiang, and thank you for presenting about the lysine acetyltransferase, KAT, in developmental disorder.
Okay, thank you. Can you see the screen now?
Yes.
Okay, I will try to be general. I will not get into the specific too much. So I will start with, let me see. Yeah, this, can you, yes. So this basically, the lysine acetyltransferase. So you have the chemically, you have this lysine structure here, and so then basically, this is enzymatic reaction. You have added this acetyl group there, and this reaction can go back and forth. So in nature, you can see this type of enzymatic reaction almost in all organisms, from bacteria to yeast to humans. So it's quite general. Ooh. Okay, so so then there, in addition to acetylation, now actually there are some other type of modification very similar to acetylation. So you see the acetylation here. So basically you have this one CH, the two carbons there. And over here, just add an extra one. So there's three, 1, 2, 3. So then 1, 2, 3, 4. So in English, it's more complicated. In Chinese, actually, we call this is just something like tri, and this is tetra. But in English, it's called Mutation, and this is called Blue Pyronylation, and then the one with 2 called the acetylation. So that's the reason. So this ester group, basically, I think everybody knows vinegar. So the structure, this is a part of the vinegar structure, and also alcohol. So we all drink alcohol, right? Take a beer, liquor, things like that. So this is here's the structure. And so yeah, so then for the lysine, basically initially was isolated from lysate, from the milk protein called casein. So this is because of the degradation. So that's why I call one of the amino acids is called the lysine. And so then for the Blue Pyronylation, so this basically Blue is like a first. And for those type of enzymatic reactions, there is one family of enzyme. Actually, in the human genome encodes about now maybe 14 or 15 such enzymes. So they can be divided into let's see. Check the participant to see. Okay. So there's like 14 or 15 of them in human, maybe in mice and in mammals. So then they divide into different families. So there's 5 of them called the Mys family, because they all share one particular domain called the Mys domain. And so you all know cat 6b, and there's a cat 5, 7, and 8. And I will tell you a little bit about cat 5, 8 as well. And the cat, among the 5 of them, cat 6a and b are more similar than the other 3. And all this protein actually are conserved from they share domain with proteins in yeast, in plant, and some of the members actually were initially identified in yeast. And so this is the first paper, and I highlight here. This is from MIT, David Hausman's lab. And in the same issue, this is September 96, same issue, there's another paper. It's back-to-back in the same journal. The Nature Genetics is one of the top scientific journal. And basically they identify a family of enzymes. One of these is called MOS, and because I think MOS stands for, it's monocytic leukemia. Zinc finger proteins, that's called MOS, monocytic leukemia. So MOS. And this actually, there's another name. It's very confusing for people not in the field. It's called also Mys3. And now we all know it's cat. Is it okay? Did I check a participant? And so then another group from UCSD, and they studied the yeast protein. They share similar, so that's similar domains with MOS. So those are the 2 papers. At that time, I was actually still a trainee at the NIH in Maryland. So we were so excited by these 2 papers. I remember we read these papers many times. This is just a hypothesis that some of this enzyme could be acetyltransferase. So then the year after, let me see the next one.
Dr. Xiang, yes, we can hear you well.
Can you hear?
Yes, very well.
Okay, so then next year I came to McGill. So then a former postdoc joined the lab, and Natalie Champagne and some other students. So basically identify another protein we call MORph, now known as cat6b, also Mys4. And so basically MORph, very similar. It's like not twins, but like sister and brothers. Yeah, very similar proteins. 6b, that's why I call it 6a and 6b now. So basically we show that this is the enzyme for acetylation. And then at the same time, this is another important paper from Tim Thomas in Australia. So basically they identify the mouse protein, now known also the mouse protein of cat6b. They call the Korkoff. It's German, I think, and means a square head. Because why is that? Because the mutant mice lack of this protein has this head. It's like a square. So basically affects the bone development and also the brain development. So basically this is one of the first indications that this protein is really important in brain development. And so yeah, the name is confusing, but the study is very important. And just to make the long story short, we have a collaboration with Cote's lab at Laval University, and identify this is a complex, basically. The Cat6A and the 6B, they need something else, some other helper like a PRPF1 to activate those enzymes. So that's why, just without this protein, Cat6A and B may not function properly in the cell. And this is two studies from a former student, Ling Ya. Basically, she made a knockout. Now she's a professor, has her own lab studying brain development. And she basically made a knockout and demonstrated that the mice, if the mice doesn't have this protein in the brain and their brain have problems, and also affects the neural stem cell, and also something called dendrogyrus. This is a place in the hippocampus of the brain. It's very important for memory and learning. So that's why it's related to intellectual development. And this is basically, it's kind of support this one paper very nicely. So you have the 6B important in mouse brain development. Now you have BRPF1 also important in brain development. So those are basically based on that. Basically, we think those proteins are very important in the brain. And one important question is. Because the early study was in biochemistry and then in mice. So then the next important question is whether there is a link in human and patients. And that basically I think some of you met Dr. Kapoor and because there's 3 studies. This was a few months apart in 8-9 years ago. So demonstrated that mutation in Cat6B basically cause different type of syndromes. So although they have different names, they overlap. Genitopatellar, this affects the neocap and the reproduction system. And also those patients have intellect problems in IQ. So those studies are, I was very interested in those studies, and basically those are also very consistent with the early studies in mice. So then the recent work for Cat6B, now I think there are about 60 or 70 patients with Cat6B mutations. This is a very new study from Dr. Kapoor's lab, collaborated with me and many others, so many authors. So this is Cat6B. So then Cat6, so here, so you have those mutations. So here you can see the exons. Okay, this is from exon sequencing mutation. You have the box, the exon. So then those lines, this is like intron. So usually if the mutation in the intron, so then shouldn't affect too much, unless it is in the boundary between intron and exon. That may affect function. Another thing we don't know yet, because in for gene, not just for the exons, there's other elements like called a promoter and some others. So we don't know whether effects those may cause similar problems. So that's, I think, one area for future studies. And then if you look at the detail of the mutation, some of you, I think maybe may get the genetic report, and you may not understand what are those. And basically, This is, for example, here, it's a DNA sequence. C is just for the coding, and this is the position of the exon, the coding sequence. So then A is like one of nucleotide changed to G. This is the DNA. Over here, the next one actually is amino acid protein. Yeah, I think it's a typo. It should be, sometimes you see just one letter. This is lysine. Then this is position change. So then you have the FS into alanine. So then you have the FS. Basically, it's called the leading frame shift. And so then, for example, this one is like histidine 767 changed to tyrosine. So in our body, there are 20 common amino acids and 4 nucleotides. You can see all those, for example, proline. This one, for example, proline to proline. Personally, I was saying this shouldn't have any effect. Okay, so sometimes we got the mutation may or may not have effect. For example, this one shouldn't. And this one, actually, we don't know, need to really test. So then this is a star, it's like a truncation, usually should affect. Okay, so this just give you an idea that sometimes you see a mutation, doesn't mean it's pathogenic. Okay, I just want to mention that also depends on position. Some of the amino acid change may not affect the function of the protein. Let me see what else. For example, this tryptophan to arginine, we don't know. But based on some analysis, for example, I think it's important. And so that's why we need somebody to test, to verify whether this, for example, this or this are really pathogenic. So I just want to give you some examples. As I said, for example, this one, proline to proline, shouldn't have any effect. And this is for the cat6b and then cat6a. Many of you are very familiar with. And this was the link to patients was made by 2 groups. I think 5 years ago. Those are very important studies published in this one of the top journal in human genetics. And this is a more later study, more comprehensive, 76. Now I guess, I'm not sure how many registered in the foundation. I guess maybe 300 or 400 individuals with a mutation, right?
Yeah, the 300 cat6a.
Yeah, something like that. So, but this is still only in Europe. I think in developed countries where exome sequencing is routine. Let's say in Middle East, in some of the developing countries, that technology is not available yet, so we don't know yet. So I will talk about that later. So this is cat6a. Yeah, so then, as I mentioned, the BRPF1, that is like a facilitator of cat6A or B. So then we and another group in France, Emily Piton and Dr. Mendel, and they actually also made a similar discovery that the BRPF1 mutation causes intellectual disability. The clinical features overlap with patients with cat6A or B, and there's a distinction also. Typically, the features are not as striking as those patients with cat6A or cat6B, because this is a facilitator, not the enzymatic engine. Okay, so this is a very new study. This earlier this year, basically we demonstrate, as I mentioned earlier, not just acetylation. So there's something called a Blue Pyronylation. Basically, this, as I said, is, the side chain has three carbons. let me just go back to. Yeah, so you have the Blue Pyronylation here. So you have the very similar to acetylation. So this is the acetylation, basically chemical. When I was student, university student, I used to study chemistry. So that's why I like the structure. So you have the ester group. So here you have the Blue Pyronylation. This is the side chain. Okay, this is one of the shortest fatty side chain. So the question is, what's the function of this modification? Not just all histones have different spots for modification. Not all those spots, only particular spot, as this particular lysine called lysine 23. And it's very specific. So that's what's the function of this modification? We still don't know. We are still trying to figure it out. But I think it's very similar to acetylation. This is also from many labs from in Europe and in US and Canada, in Israel also. And Dr. Kapoor is major collaborator. And I mentioned the Cat6A, B, BRPF1. So then in the family, there are 3 other cat acetyltransferases. Cat8 is one of them. This is a new study also we just published with collaboration with many labs, including, so she is from Israel, and some of them from Europe, Kapoor here. So basically Cat6A mutation also causes intellectual disability, epilepsy, and some other issues. But overall, it's milder than patients with Cat6A or Cat6B. Actually, in one case, we had a family, actually, some of the, I think one of the parents actually doesn't have anything symptoms, but I think yeah, his kid had some issues. So this is Cat6A, and also we carry out the mutation in mice, and those mice actually we show that it's very important in brain development. So this is very consistent. And then for epilepsy, I think some of the compounds were used in some of the patients and were quite effective. And then the last 2, one of them is Cat5. And this is from Dr. Kapoor's lab, and basically shows that Cat5, I think in this study there are 3 patients. And in this one, there were 9 patients. And this is 3 patients. The clinical feature overlap with those I mentioned earlier, except those 3 patients have huge issue with the cerebellum. So that's important for control movement and sleep disturbance. And also those the 3 kids have behavioral problems, and they tend to major issues with behavior. They tend to be getting angry much more easily, things like that. And has epilepsy also. Okay. So then for Cat7, we don't know yet. So we are studying to see what's the issue. So then If we look at back, one of the important questions is where did the mutation come from originally? So, so we call some called de novo, and some are inherited. Even for the inherited, so there must be the mutation where they inherited, right? So must be from also occur somewhere in the first place due to spontaneous mutation. I think this is actually mainly, I think, due to normal DNA replication error even in normal human cells and in germlines. And so then this kind of low rate replication error, so then cause spontaneous mutation. So usually you will see like maybe 10 or 30 spontaneous mutations per generation. And this occur rather randomly. And if you consider the genome size, we can calculate the frequency. And you see a slightly more higher rate in mutation rate in male than female. And from that, we can estimate how many cases around the world. For example, we can use 6A as an example. And so 1 to 30, let's say we take one number from the middle, 15 mutation, let's say. So then Cat6A, the coding sequence, just the coding sequence, exons, if you add together, I think it's around 6,000 nucleotides. The protein is 2,000, times 3 is 6,000. So then the genome size is 2.2 billion, and the total population in the world is 7.8 billion now. This grows very fast. I'm quite concerned about this. Just 10 or 20, 30 years, only 4 or 5 billion. So this is anyway, that's not our topic today. So if based on this calculation, you get about 10,000, 100,000 individuals with Cat6 mutation. As I mentioned earlier, some of the mutation may be silent. It won't have any problems. So maybe if you look at the amino acids, not all of them are important. Some only the important affected, so then cause problems. So about 10%, I would say, this is estimation. So this will give you about 10,000 clinical cases. This is one way to estimate. So then we can also estimate this from another way. So now, as I think I mentioned that 300 or 400 cases identified already. I think the foundation has around 300. Let's say if 400 cases for Cat6 in Europe, US, and Canada. And then this population there is like 1.1 billion. So then times the total population. This gave about 3,000 cases. So this is only, we're talking about from exome sequencing. So this only covers the exons. Exon added together is called exome. So then for the gene, there are other elements. There's something called a promoter, regulator. We don't know yet. So based on this, I would think maybe around 5,000 cases around the world. Okay, so this gives you some idea how many cases around for Cat6 in the world. So then you add other genes together. So then you have 4 or 5 members and different. So then yeah, you get some idea how many cases total in the world. So overall, this is a very, very rare disease. Okay, it's very rare. And so then that's just for the mutation. So then at which point the mutation may occur? So this is, we have to look at the development of, so you have 2 parents provide sperm and egg. So then form together, this is something we call zygote. So then develop to embryo. So gradually So during embryo development, there's something called designated certain population as germline. So that's for generation of future generation, provides sperm and egg. So then fetus, baby, and become new parent and go back to this cycle. So this is the germline. So basically anywhere. So could it be from the parents, or could it be sperm, or could it be egg? Especially sperm, could it be a mixture? So then in our, if I'm sure if you check, ask for genetic diagnosis, they'll ask for some, let's say, blood. So that's somatic. So then, so basically we mainly isolate somatic tissues for the mutation. So we don't know germline tissues, whether any mutation in sperm or eggs. And in a zygote stage also, then the embryo development, so then from here to the germline, so there are many steps. So anywhere that could happen. So that's why, sometimes it's called de novo, not in the parents, but in the baby. But it still could be from the sperm or egg. So then in that case, So de novo, how basically it depends on how you define the term. Okay, this is giving you an idea, So all this, as I said, this mutation occur rather random. Okay, so this is just that important point. I think it's quite important for parents, who have kids, because some parents may try to figure out, why, these things like that. This is just sometimes random. It's nobody's fault or anything. It's just that's the nature. Okay, so that's I want to tell you. And so then, how the mutation affect development? I think if you look at the early paper, you can see some of the paper claim, the mutation is dominant. But personally, I think it's more like something called a haploinsufficiency. So basically, let's say usually it's heterozygous, right? 50% is affected. So then this 50% is not sufficient. That's called haplo. Haplo is like, because our genome is diploid. It's like 2 copies. Just 1 copy is not enough. That's called haploinsufficiency. You have the 3 parts. Haplo is like 1 copy, and in is not, and insufficiency. So it means 1 copy is not enough, or 50% of the gene is not enough. So I think this is the major mechanism. And yeah. Any questions? Okay, we can move on. So then inherited, mainly I think it's de novo. Usually you don't see in parents, but in a few cases you see in parents. And as I said, it's mainly de novo, but sometimes we see parents, let's say blood has a mixture of the normal and mutation. It's called a mutation. Genetic, or could it be germline? So this is more challenging.
That I cannot go with her on
This
I'm
is
only going tomorrow. That's what she said. That's what she said. Ow! Ow!
No kicking.
Is everything okay?
Get in your bed.
Yeah, she did. So you are. Go.
Okay. Related to germline, was it? This is actually this case from Japan illustrates this point very well. So you have the
Dr. Young, can you mute the microphone? There is one microphone that is.
Can you hear?
Yeah, you can right-click on the name and mute it.
Oh, okay. Who?
I think it's David.
David? Okay. Yeah, I did it.
Okay, thank you.
So you have this actually, it's a quite a unique case. So you have the father, the DNA sequence here. So you have the DNA sequence is basically different peaks. The color is pseudo color. So basically we give a T, red, green is A, G is dark, and this is blue, C. So over here, this is basically here. So this is one of the patient, one of these three. You can see that it's a mixture of C and T. So basically one copy of you have two copies of the gene, right? One copy is mutated. So C changed to T. And this position, this C dot, the first C dot is small c. That means coding sequence of the gene. The position 30, 70. So this is a 37 divided by 3 is like 1,000 along the first 1/3, a junction of the first 1/3 and the second 1/3. And so this is basically changed to this star. It's introduced a stop codon. So normally it's CGA, now changed to TGA. It's a stop codon. So it's just unlucky. Okay, so then you have two other, have the same one. So then the striking thing is both parents, father and mother, are normal. But we should say, if you look at the paper, this is blood samples. I think maybe there is something called germline mutation. Basically, that's the conclusion that the authors made. It's parental germline mutation. I think this is a very unique case. So then the question is this from the mother or father? That's a scientific question. And so as a scientist, we would love to know that. But so then I talked to the Dr. Yoshiura, and he mentioned that the parents didn't give the consent to do that because we will have to check the germline, basically check the. I think for women it's more difficult. For men it's easier. We just check the sperm mixture. But if that usually you need to get a consent, so then that's raise an ethical issue. The question is really for the family. Do the family really want to know whether it's a father or from mother? I think it's for them. I think they prefer not to know. So there is a different view from science and ethics. So this is a quite interesting example. So yeah, I think it's for them. I think I fully understand they wouldn't want to know because yeah. Okay, so this is a unique example. 3 brothers. Usually, I often tell parents when they talk to me, they have one kid, for example, had mutation. So they will ask me whether the chance for the second kid have the same mutation, how high. I would, this is like, because it's random, right? So the chance is very low unless, there's a germline mutation, basically either a sperm or egg have a mixture. So, but this is usually it's very, very low. So far I only found this case. Okay, so then normally, let's say, yeah, just to be sure. So then if parents want to have a second child, so then this we can always check it out whether it's true. Whether, there's such thing called germline this. I think that's basically, let me see, yeah. So then I mentioned Cat6A, Cat6B, and they are facilitate or activate BRPF1, or the mutation in each of them can cause problems in development. So then I also mentioned the Cat6A and Cat5. So then all this basically provide a scientific explanation for the reason, give scientific explanation for the disease in the individuals who have the mutations. So then the next question is how can we manage this kind of problems? So for genetic counseling, so there, as I mentioned, there's also ESCA issue, and as I believe in the future, it's still should be possible to do prenatal analysis, but that also raises an ESCA issue. So then the next question is treatment. For treatment, could it be its development? It's more challenging, but as yeah, its development it's more challenging. But there's some issue. For example, for the brain development, as I mentioned earlier, neural stem cell is one of the place where there's problems. So let's say during early development, if we can give patients normal neural stem cells, so that's a potential option. So this can be first tested in mouse models to see whether this kind of approach will work. So that's one way. Another way is use chemical compounds, and luckily, so this go back to the chemistry, the first slide. Here you have the acetylation. So basically, so Cat6A, for example, 6B or other, if you lost one allele, so basically this reaction is not enough. So then you have this the reverse reaction. For this called the deacetylase, there are a lot of inhibitors now. So if you block that, so then can partially compensate that. Actually, some of the drugs, like epileptic drugs, are actually like inhibitor for those enzymes. So then can indirectly boost the function of the acetyltransferase. So that's another way to go. So the problem with this is that those inhibitors usually, because those are not specific, so may have a side effect. So that's the potential problem. Yeah. So yeah, that's basically this family. So starting from pure biochemistry, as I mentioned, the first two study, one is from analyzing a mutation in leukemia from MIT, and another study from UC San Diego, studying basically yeast protein. So eventually led us to the understanding of genetic disease in patients. You can see science sometimes it's there's something called serendipity. So when we started so many years ago, we had no idea that those could lead to direct connection to genetic disease. So then you could also see that scientific development advance usually when we go to supermarket, oh, we want to buy this, that, we have a good plan. Scientific study, sometimes we don't know actually where we're heading. Actually, we just try to see. For example, when we started, I was just curious, what is that? So then, from curiosity. Okay, I will stop here and take some questions if you have any.